Imagine a city that has been, under siege for months.
Right, like a really desperate situation.
Exactly. And you're the mayor, and your absolute ultimate defense strategy is to hire this elite, highly trained force of security guards.
The best of the best.
Yeah. So you equip them, you station them on the high walls, and finally, you can sleep soundly.
Makes sense.
But what you don't realize is that the invading army, well, they didn't bring battering rams or, siege towers.
Oh, wow. What did they bring?
They brought briefcases full of cash.
Ah, I see where this is going.
Right. They have systematically and quietly bribed your elite guards. So now, the very people you hired to protect your city, they're actively opening the gates.
Wow.
They're handing out maps and basically ensuring the enemy takes over without a single alarm ever being tripped.
It is, it's the ultimate betrayal from within.
Completely.
And unfortunately, it perfectly illustrates the exact biological strategy deployed inside the human body by one of the most lethal malignancies we face today.
Well, welcome to the deep dive. Our mission today for you listening is to decode a highly complex, brand new piece of medical research published in March 2026.
Yeah, and it's a fascinating paper.
It really is. It unravels this long-standing mystery surrounding hepatocellular carcinoma, or HCC, which, is the most common form of primary liver cancer.
Right, highly lethal.
Very. And we're going to explore how a specific molecule acts as this corrupting mastermind, basically running that exact bribery scheme to help tumors evade the immune system.
And what makes this research so compelling, I think, is really the architecture of the evidence.
What do you mean by that?
Well, the researchers didn't just find a correlation and, publish it. They built a case like forensic detectives, grading the evidence step by step.
Oh, I love that. Like an investigation.
Exactly. They started in silico, which just means they mined these massive global databases of patient records to look for macro-level patterns.
Right, the big picture.
Yeah. Then they zoomed in using incredibly precise single-cell algorithms to find out exactly which cells were involved.
Okay.
From there, they used spatial mapping to identify the physical locations of these cellular crimes. And finally, they proved their theory in real-world lab and animal models.
So they went all the way from databases down to living animals. That's huge.
It is the gold standard of biological proof.
So to understand the crime, we first have to really understand the scene of the crime, right? In oncology, this is known as the tumor immune microenvironment, or the TIME.
Yeah, the TIME. And going back to your analogy, the tumor immune microenvironment is our city under siege.
Okay, lay it out for us.
For a long time, we thought of a tumor as just a chaotic lump of mutated cancer cells, just growing randomly.
Right, just a mass.
But it's actually a highly complex, almost self-sustaining ecosystem. It hijacks local blood vessels for nutrients, it builds structural connective tissue, and crucially, it is packed with immune cells.
Wait, immune cells are inside the tumor?
Oh, absolutely. In a healthy body, your immune system should, recognize cancer cells as abnormal and just destroy them.
Right, that's their whole job.
But inside the TIME, the tumor essentially neutralizes the local immune cells. It bribes them to stop fighting and start actively supporting the cancer's growth.
And according to the data in this paper, the security guards who take the biggest bribes are a highly specialized group of cells called tumor-associated macrophages, or TAMs.
Yeah, TAMs. Macrophages are totally fascinating. They are usually the frontline soldiers and kind of the cleanup crew of your immune system.
Right, they literally eat the bad stuff.
Exactly. They patrol your tissues, swallowing up cellular debris, dead cells, foreign invaders, you name it.
Sounds like a good guard to have.
But their defining characteristic is their plasticity. I mean, they can radically change their biological function based on the chemical signals in their environment.
Okay.
Broadly speaking, we divide them into two main operational states. We call them M1 and M2.
And for you listening, I actually have a little mnemonic device to help keep these two distinct, because honestly, it's vital to understanding how this cancer survives.
Oh, I'd love to hear this.
Think of M1 as murder one.
Ha! Murder one. I like that.
Right. These are the aggressive, pro-inflammatory macrophages. They are actively fighting, releasing toxins, and murdering the cancer cells.
Exactly. That's the state we want them in.
But M2, on the other hand, is medic 2. These are the corrupted guards.
Medic 2. That's a great way to remember it.
Yeah, because their normal job is to calm down inflammation and heal damaged tissue after an infection is over. But the tumor tricks them into doing this inappropriately.
Right. They start treating the tumor like a wound that needs healing.
Exactly. So these MEDIC2 cells start building new blood vessels for the tumor, they repair its damaged tissue, and they suppress the rest of the immune system. They basically throw a protective cloak over the cancer.
That analogy hits the nail on the head. In aggressive liver cancer, the tumor environment is overwhelmingly dominated by those M2 medics.
So they're just completely bought out.
Completely. And the million-dollar question in oncology has always been, how exactly does the tumor force that switch? What is the actual mechanism of the bribe?
And that brings us to the primary suspect in this new paper, a molecule called MARCKS.
Yes.
MARCKS. It stands for myristoylated alanine-rich C kinase substrate.
Yeah, let's just stick with MARCK.
Yeah, let's definitely stick with MARC.
What's deeply counterintuitive here is what MARCS normally does.
Right, it's not usually a bad guy.
Not at all. Historically, biologists considered it a purely structural protein. It's a cellular architect that manages the cytoskeleton.
The cytoskeleton, like the cell's bones.
Sort of, yeah. It's the internal scaffolding of actin filaments that gives a cell its physical shape and allows it to move around. It was never really thought of as an immune regulator.
But in the context of HCC, this innocent architectural protein has gone entirely rogue. It is the mastermind handling the physiological bribe money. So, if we pause and look at the board we've set up, let's do a quick sub-summary here, kind of a bullet point recap for you listening.
Good idea.
Okay, bullet 1. We have a deadly liver cancer, where the tumor successfully bribes the immune system's frontline guards.
Bullet 2. It turns them from M1 murderers into M2 medics.
Right. And bullet 3. The key operator making that transaction happen is a structural scaffolding protein called MARCKS.
Perfect recap. It's a brilliant survival strategy from the cancer's perspective, hiring the enemy's security force.
It really is. So my question to you listening is this. If you were a cancer cell, wouldn't your absolute best survival strategy be to hire your enemy's security force?
It makes total evolutionary sense.
It does. But to see how devastatingly effective this is, we have to transition into the next phase of the deep dive. We have to look at the actual patient data.
Right, because we need to know what this looks like in the real world in actual humans.
Exactly. So to do that, the researchers analyzed the TCGA-LIHC database,
which is this massive repository from the Cancer Genome Atlas, specifically focused on liver cancer patients.
Right. They wanted to see if the presence of this MARCKS protein actually changed clinical realities for these people.
And the numbers are, well, they're staggering. Patients with high levels of MARCKS expression in their tumors had drastically worse clinical outcomes across every major metric.
Let's break those metrics down because they're important. First, they had significantly lower overall survival, or OS.
Right, which simply means the total time a patient lives after diagnosis, regardless of the cause of death, it plummeted.
And they also had worse disease-specific survival, or DSS.
Yeah, meaning the cancer itself was the direct cause of mortality.
And finally, much shorter progression-free intervals, or PFI.
That's a critical one. PFI measures the time before the cancer aggressively starts growing again after a treatment. For these MARCKS high patients, that time was severely compressed.
So the cancer just rushes right back.
Exactly. It's relentless.
And beyond just survival timelines, the specific clinical manifestations of these MARCKS high tumors were severe.
Very severe. These patients presented with highly elevated alpha-fetoprotein, or AFP levels.
Let's explain AFP, because that's a big red flag, right?
It is. AFP is a protein normally produced during fetal development. In healthy adults, it should be practically undetectable.
But in these patients,
it was spiking massively.
Yeah.
In this case, well over 400 nanograms per milliliter, which indicates a highly dedifferentiated, primitive, and aggressive cancer.
Basically, the cells have forgotten what they're supposed to be and are just multiplying wildly.
Precisely. They also saw higher rates of vascular invasion.
Which is when the tumor physically breaks into the local blood vessels, right?
Yeah, to metastasize and spread. Plus, they saw higher histologic tumor grades, meaning the cells looked completely chaotic and highly mutated under a microscope.
But looking at the source material, there's a fascinating genomic split that happens here.
Oh, this part is incredible.
Yeah, the data shows that RRCKS expression actually divides the patients into two completely distinct genetic subgroups. If a patient was in the RRCKS high group, their tumor was predominantly loaded with mutations in a gene called TP53.
And that is a critical mechanism to understand. TP53 is, it's essentially the master emergency brake on cellular division.
The emergency brake. I like that.
Yeah, it monitors the DNA for damage, and if it finds mutations, it stops the cell from dividing until the DNA is repaired, or if it's too far gone, it forces the cell to self-destruct.
So when the TP53 gene is mutated, basically when that brake line is cut, the cell divides recklessly, passing on all that damaged, unstable DNA.
Exactly. Now contrast that with the RRCKS low group, where the tumors were dominated by a totally different mutation pathway involving the CTNNB1 gene.
So what does that actually tell us?
What this tells us is that high RCK expression isn't just some random consequence of the cancer. It is a defining integral feature of a specific, highly aggressive genetic blueprint. It clusters precisely with tumors that have completely lost their TP53 emergency brakes.
That makes a lot of sense. All right, we're another 5 minutes in. Let's do our next bullet summary to lock this in for the listener.
Sounds good.
Bullet 1. MRCKS is heavily overexpressed in aggressive liver cancer.
Bullet 2.
Yeah.
It is an independent predictor of terrible clinical outcomes like low survival and high AFP.
Right. And bullet 3. It defines a unique genetic fingerprint clustering with those broken TP53 tumor suppressor genes.
Perfect. But I do have to point out that while this bulk clinical data gives us a really powerful macro view, it has a major limitation.
Okay, what's the limitation?
When you analyze a bulk tumor sample, you are basically grinding up millions of cells, cancer cells, immune cells, structural cells, and you're sequencing them all together.
Like a smoothie.
Exactly. We can see that MRCKS is highly expressed, and we know it correlates with terrible outcomes, but it's like, imagine hearing a massive, deafening roar in a crowded stadium. You know it's incredibly loud, but you have absolutely no idea who is actually doing the shouting.
Ah, I see. So here's a question for you listening.
Yeah.
How do you isolate the single corrupt voice in a crowd of billions of cells?
It's a massive challenge.
To do that, we have to transition into segment 3. We have to dramatically zoom in.
Right. Because the bulk data couldn't answer the who and the where, the researchers deployed 2 of the most advanced techniques in modern molecular biology.
And those are?
Single-cell RNA sequencing and spatial transcriptomics.
Let's unpack those.
Right.
A great way to visualize this combination is to think of it as like Google Earth for tumors.
That's a great analogy.
Yeah, bulk sequencing is like looking at a whole city from space at night. You can see a massive glow of electricity, but that's it.
Right.
But single-cell sequencing lets you zoom all the way down to the street level to see exactly which individual houses have their lights turned on. That gives us the precise who.
Yes, and then spatial transcriptomics takes it a step further by giving us the exact GPS coordinates of those illuminated houses within the city grid. That gives us the where.
Okay, so let's look at the single-cell discovery first. The who.
The researchers used algorithms to separate the tumor ecosystem into 22 distinct cell clusters. They looked at the actual liver cells, the T cells, the B cells, the blood vessel cells, everything.
And where was ARC-RCS hiding?
What they found was that MARCKS was not evenly distributed at all. It was highly enriched in the myeloid lineage. Specifically, it was heavily concentrated inside those exact immunosuppressive tumor-associated macrophages.
The M2 medics we discussed earlier.
Exactly. The rogue architect protein is found specifically inside the corrupted guards.
But wait, that raises kind of a chicken or egg scenario, doesn't it?
How so?
Well, did the MARCKS proteins show up after the macrophage was already corrupted, or is the rise of MARCKS actively driving the corruption?
That is a crucial distinction. And to untangle that, they use these computational tools called cytotrace and pseudotime.
What do those do?
These algorithms act like a form of biological time travel.
Time travel?
Sort of, yeah. Because you can't watch a single cell evolve under a microscope for weeks, these tools analyze the subtle differences in RNA across thousands of cells to construct a mathematical timeline of cellular development.
Like a flipbook of how the cell changes.
Exactly. They can map exactly how an innocent precursor cell, which is called a monocyte, matures over time.
And the trajectory they mapped is incredibly revealing. As these innocent monocytes march down the developmental timeline to become corrupted M2 macrophages, their internal levels of RRCKS steadily and dramatically rose.
And crucially, RRCKS didn't rise alone. No, it ramped up in perfect lockstep with a whole suite of specific genes that shut down the immune response. Genes like CD163, MRC1, KLF4, and HAVCR2.
Those are a lot of letters. What do those genes do?
Basically, they are exhaustion and M2 markers. They are genes that tell the cell to stop fighting, start repairing, and essentially go to sleep.
So the chronology here is the key. MARCKS in vial is not just a passive marker of a corrupted cell. Its production actively ramps up at the exact moment the macrophage is transitioning into its immunosuppressive state.
Yes. It is intimately involved in the act of taking the bribe.
But understanding who is corrupted is only half the battle. The spatial mapping, the where, is what reveals the true defensive strategy of the tumor.
Right, going back to our Google Earth GPS coordinates.
Using spatial transcriptomics on clinical tissue samples, they mapped the physical locations of these MRCAS positive macrophages, and they discovered this profound bad neighborhood effect.
The spatial data proved that these corrupted macrophages are not just, floating randomly through the liver tissue.
Where are they?
They purposefully, physically cluster together in highly specific zones around the tumor. And when you look at who they choose for neighbors, it's a who's who of immunosuppression.
Like a gang gathering in one spot.
Exactly. They group up with exhausted T cells that have lost their ability to fight, regulatory T cells, or TREGs, whose entire job is to secrete chemicals that calm the immune system down.
And cancer-associated fibroblasts, right?
Yes, CFs, which build dense physical scar tissue.
Wow. So we aren't just looking at isolated corrupted cells. We are looking at fortified, coordinated neighborhoods. The tumor is using these RRCKS positive macrophages to build physical and chemical walls that completely block normal, healthy immune cells from ever getting close to the cancer.
It's an incredibly sophisticated defense mechanism.
Okay, let's do our third recap. We are diving deep here.
Bullet 1, single-cell data proves RRC test specifically targets the myeloid macrophage populations.
Bullet 2, trajectory algorithms, like that pseudotime flipbook, show RRCS rising precisely as macrophages become corrupted.
And bullet 3, spatial mapping proves these RSS-positive cells live in heavily guarded bad neighborhoods that block normal immune responses.
So if we summarize, we have motive, we have the suspect, and we have their exact location on the map. We have a mountain of circumstantial evidence.
We do, but correlation, no matter how strong, is not causation.
Exactly.
Yeah.
Here's the question for you listening. If we know they are the scene of the crime holding a weapon, how exactly are they pulling the trigger?
Right. How does a scaffolding protein, something meant to build cellular structures, actually reprogram the mind of an immune cell?
To bridge that gap, to go from algorithms to absolute causation, we have to move into our final segment. We have to go into the laboratory.
The researchers needed to find the biological mechanism, the physical chain of events, so they utilized a tool called CellChat.
CellChat sounds like an app.
It kind of acts like one for biologists. It analyzes receptor and ligand pairs to map out exactly how different cells are communicating with each other.
Okay, and what did they find?
This analysis revealed that the cellular telegraph system mediating this corruption is the JAK-STAT3 signaling pathway.
The JAK-STAT3 pathway. Let's explain that. It's a fundamental chain of proteins that transmits information from the outside of a cell all the way into the nucleus, right?
Yes. It tells the cell's DNA to change its behavior, often instructing it to grow, survive, and in the case of macrophages, suppress inflammation.
But let me push back here for a second to clarify the mechanics.
Sure.
We established earlier that MARCKS is highly concentrated inside the macrophage. So is the macrophage using its own RCKS protein to corrupt itself? Or is the tumor cell producing RCKS and using it to send a corrupting signal outward?
That is a brilliant distinction, and it's actually the latter. The tumor cell itself is the source of the signal.
Okay, so how do they prove that?
To prove this, the researchers performed a genetic knockdown. They used small interfering RNA, or siRNA.
Let's define siRNA. It's basically like a microscopic hitman, right?
Yes. It's a technique where you introduce custom RNA strands that intercept and destroy the messenger RNA blueprint for a specific protein before it can even be built.
Right.
So they use this siRNA to completely silence the MARCKS gene inside human liver cancer cells.
And when they silenced MARCKS in the tumor, what happened to the signaling pathway?
The activation of STAT3 in the surrounding cells plummeted.
Wow.
In molecular biology, proteins like STAT3 are activated by phosphorylation, essentially, attaching a phosphate group to the protein. Think of it like turning a key in the ignition.
Okay, the ignition key.
Without that key, STAT3 just sits dormant in the cytoplasm. When they removed MARCKS from the tumor, the ignition key for STAT3 in the surrounding environment was never turned. The pathway just went completely dark.
And the cascading effects of shutting down that pathway were immediate. In the Petri dish, the cancer cells whose MARCKS gene was silenced simply stopped thriving. They couldn't form new colonies.
Right, and their growth just halted.
And when researchers created an artificial scratch or wound in a layer of these cells, the silenced cancer cells lost their ability to migrate across the glass to close the gap. Their structural mobility was broken.
But, a flat piece of glass is not a living organism. So they escalated the evidence to animal models. They injected these human liver cancer cells under the skin of mice to observe tumor growth in a living system.
What happened to the mice?
Well, in the control group, where a MARCKS was functioning normally, the xenograft tumors grew rapidly and aggressively.
As expected.
But in the mice where the MARCKS gene had been knocked down, those tumors were dramatically smaller, lighter, and physically shrunken.
That physically proves that the tumor needs RCKS to grow. But it still doesn't fully explain the bribery of the immune guards. How does the tumor actually transmit the bribe?
And that leads us to what is arguably the most elegant experiment in the entire study, the co-culture mind control experiment.
Oh, I love this part.
This experiment directly proves the mechanism of cellular reprogramming. The researchers took a population of standard lab-grown macrophages that essentially act as blank slates.
Totally innocent cells.
Totally innocent. Then they exposed these blank slate immune cells to something called conditioned medium.
Which is what exactly?
Conditioned medium is the nutrient-rich fluid that cancer cells have been living and growing in for days. It is essentially a chemical soup, absolutely saturated with whatever secret proteins, cytokines, and signaling molecules the tumor cells have been broadcasting into their environment.
So they take this tumor-conditioned fluid from highly aggressive liver cancer cells and just bathe the innocent blank slate macrophages in it.
Yes. And the result, the macrophages immediately transformed into corrupt M2 medics.
Unbelievable. The chemical soup alone, without any tumor cells actually present, was enough to brainwash them into upregulating their immunosuppressive genes.
Exactly. But here's the critical pivot of the entire study. They repeated the exact same experiment, but this time they used conditioned medium from liver cancer cells where the MARCKS gene had been genetically silenced.
And the mind control completely broke.
Completely.
The fluid from the silenced tumors had no effect. The blank slate macrophages bathed in this new fluid did not transform into M2 medics.
Because their STAT3 ignition keys were never turned and they remained normal.
This definitively proves that without the RRCS protein, the tumor is biologically incapable of broadcasting the corrupting signal. The bribe money never gets delivered.
It's an airtight case.
All right, our final recap.
Bullet 1. The biological weapon is the STAT3 pathway, which drives the tumor's protection.
Bullet 2. Removing MARCKS in living cells stops cancer growth, migration, and invasion.
And bullet 3. Tumors absolutely need RRCSS to secrete the chemical signals that brainwash macrophages into the M2 state.
Which brings us to the ultimate question for you, the listener. Why does knowing the microscopic habits of a seemingly boring scaffolding protein matter in the real world?
That's the most important question.
It matters immensely because of how we currently attempt to treat advanced liver cancer. Right now, the most advanced frontline therapies are immune checkpoint inhibitors, or ICIs.
Yes, ICIs are revolutionary drugs designed to cut the biochemical brakes off your own immune system, supercharging it so it can recognize and slaughter the cancer cells.
But the clinical reality of immune checkpoint inhibitors is plagued by a massive hurdle.
Resistance. Unfortunately, yes. For a significant portion of patients, these miraculous drugs either don't work at all, or they work briefly before failing. The tumor figures out a way to hide again.
And what this research clearly demonstrates is that MARCKS is the architect building the invisible shield that renders tumors completely resistant to immunotherapy.
By isolating the MARCK-STAT3 axis, this paper doesn't just solve a biological mystery. It provides a blueprint for a brand new therapeutic weapon.
Right. If pharmaceutical researchers can develop a targeted drug that inhibits the MARCKS protein, we could theoretically strip away that chemical fortress.
We could stop the bribe from ever being paid.
Exactly. Forcing those corrupted M2 medics back into functioning M1 murderers and making our existing immunotherapies devastatingly effective for patients who currently have no other options.
It represents a fundamental shift in how we approach the tumor environment. And I think there's a profound, broader implication here to leave you with.
Let's hear it.
For decades, the entire scientific community largely dismissed MRCS. It was viewed merely as a structural protein, a mundane piece of cellular scaffolding, like the literal beams and drywall of the cell.
Just holding the building up.
Right. But this rigorous, multi-layered research proves it is secretly moonlighting as a master immune reprogrammer. So it forces us to ask a deeply provocative question.
It is.
What other boring structural elements of human biology, the passive architectural components we completely overlook, are actually pulling the strings and orchestrating the deadliest diseases we face?
It is a slightly unsettling thought, honestly, but it's incredibly exciting for the future of discovery. It means the cures we are desperately searching for might be hiding in plain sight, encoded within the basic cellular structures we thought we already fully understood.
Exactly.
Thank you for joining us on this deep dive. Keep your curiosity sharp. Always question the architecture of the information around you. And remember, sometimes it's your own security guards you really need to watch out for.